A-Level Biology 9700, Paper 4, Variant 2, October, November 2024. Question number 1 and 2. First of all, question number 1 is figure 1.1 is a diagram of part of Bowman's capsule and glomerular capillary. So identify structures A and B. Students, you know the Bowman capsule and glomerulus or glomerular capillary. They are part of the nephron. They are part of nephron. And here you can see the Bowman capsule is a cup-shaped structure. In this way you can see. So it is a cup-shaped structure. So this cup-shaped structure is Bowman capsule. And inside the Bowman capsule, you can see glomerulus, a tuft of cilia-like structure, glomerulus is there. And here the ultrafiltration takes place. As the blood enters into the glomerulus, fine filtration takes place over here, and then filtrate is moved towards the second part of this nephron. So these are this diagram shows the part of this Bowman capsule and glomerular cavity. And here you can see inside them, just like this, you can see these are the capillaries where the fine filtration takes place inside. And here, podocytes. Sites are there. So the A structure is just like amoeba. It shows that it is podocyte. And B is endothelial. Cell. And this podocyte, they are also known as visceral epithelial cells, and they are located in the glomeruli of the kidney nephrons, and they make the lining of outer surface of the glomerular cavity. So you can see their shape, and in shape, with the help of this shape, we can easily assess that these are the pseudopodocytes. So second is the endothelial cells, which form a single layer lining of blood and lymphatic vessels, and their function is to control the opening and closing of arteries, which in turn determines blood pressure and how hard the heart has to work. So in this way, you can see these are the part of the excretory system, part of the nephron. So podocytes and endothelial cell. Now we are moving towards the next question. For your convenience, you know, already written the answers, but I will explain all this. The glomerular filtrate is produced in the Bowman capsule by the process of ultrafiltration. State the conditions required for ultrafiltration. First of all, I would like to explain this. What is ultrafiltration? Already told you that in the Bowman capsule, you can see in this way, a tuft of cilia is there, which is glomerulus, which acts as a filter, and when blood enters into this Bowman capsule, it will be cup-shaped and it will receive the filtrate after this fine filtration or ultrafiltration. So ultrafiltration means the fine filtration, that is the blood is filtered over here. So we can say that this process relies on the pressure difference to force fluids and small molecules from the blood into the Bowman capsule, forming the glomerular filtrate. So the after filtration, the blood with high pressure enters over here, then ultrafiltration takes place, and after ultrafiltration, the filtrate enters over here into this second part of this uh, nephron. So here filtrate filtrate will enter. So I would like to say that firstly the blood enters into the glomerulus and then it is filtered, which is called as ultrafiltration or fine filtration. So process of fine filtration, you can say uh, there is needed a pressure difference. So due to high blood pressure, which will be in the glomerulus, you know how this high blood pressure is created. You can say the efferent arteriole. Efferent arteriole, you can say, which brings the blood, efferent and efferent. So these are the efferent, and other one is the efferent. So efferent blood arterioles, which brings the blood into this glomerulus. So it has a larger diameter than the efferent arteriole, which that vessel which taking blood away. So it creates a high hydrostatic pressure within the glomerular capillary. So this pressure is the main driving force for filtration. It means first of all, the hydrostatic pressure. So this is the first condition, because when the blood will enter into the nephron, there should be the high blood pressure, because if pressure is not there, the fine filtration or ultrafiltration will not take place. So for fine filtration or ultrafiltration, first condition which should be fulfilled, that is the hydrostatic pressure. So hydrostatic pressure is created when there is the difference in the diameter of efferent arteriole and efferent arteriole. Efferent arteriole, when by which the blood enters into the glomerular cavity on the glomerulus, and the efferent when blood leaves it. So there is a hell of difference between these two, between these pressure of these two blood vessels. So in this way, the this pressure which is created over here is the main driving force for filtration. And the second thing which should be fulfilled at that time for this ultrafiltration, that is the permeability of the glomerular capillaries. Permeability. Permeability of the marular capillaries. Permeability will allow or not allow to enter or leave the substance in. So here permeability, glomerular capillaries have fenestrations, or you can say the small pores in their walls, making them highly permeable. So this allows water and small solutes to pass through easily. So filtrate will be formed when permeability will allow them. If it is permeable, so all important molecules, they will be filtered down into the filtrate. So they will move, they will filtered into the into this filtrate. So this is the second condition which should be fulfilled. That is, it should be permeable. So permeability will allow the molecules to move. So filtrate and all these molecules will pass, or the substance will pass into this filtrate in this space. So and next, which we can say that opposing pressures, while the high hydrostatic pressure in the glomerular favors filtration, there are also opposing pressures should also be there. So then we can say the capsular hydrostatic pressure. The fluid already present in the Bowman capsule exerts a pressure that opposes further filtration. Next, we can say the capsular osmotic pressure. This is also, we can say, a condition which can cause this fine filtration. So capsular osmotic pressure is produced the fluid which is already present into the Bowman capsule. So we can say that a balance of pressures within the glomerular hydrostatic pressure being greater than the opposing colloidal osmotic and capsular hydrostatic pressure. So we can say the balance of these hydrostatic pressure and the capsular osmotic pressure, they keep a balance. So due to this balance, it is very helpful for the filtrate to filter down, or you can say the fluid or the substance filter down into the in the form of filtrate into this Bowman capsule. So permeability, it will allow the molecules to move to in the form of filtrate. So these are the conditions which are helpful for this ultrafiltration. Now I would like to read the answer which I have written. The conditions necessary for ultrafiltration are primarily related to pressure differences and the structure of the filtration barrier. Structure of the filtration barrier. So the efferent arteriole has a larger diameter than the efferent dam. So there should be dam. Efferent arteriole, this creates a high hydrostatic pressure within the glomerular capillaries, which is the main driving force for filtration. So the Bowman capsule has a permeable inner layer and allow water and small solutes to pass through easily. Already told you, first of all, the hydrostatic pressure, which is produced at that time due to the diameter difference of the efferent blood vessels and the efferent blood vessels. So this hydrostatic pressure is produced. Secondly, the permeability. So here you can see it creates hydrostatic pressure, and then the capillaries, which is the main driving force, and the Bowman capsule has a permeable inner layer. So permeable inner layer is there, due to which the water and small solutes pass through this easily. Thirdly, the capsular osmotic pressure, which will help to keep the balance. It will keep the balance in the filtration. So too much filtration uh, can cause other issues. So it helps to keep the balance in the filtration process. So these are the conditions. Other conditions, we can also discuss the other conditions, but the marks are only two. So we will, it is NF40, your marks. Now I'm moving towards the next question. As the newly formed glomerular filtrate, now after filtration passes along the proximal convoluted tubule, selective reabsorption takes place. The fluid remaining in the proximal convoluted tubule eventually forms urine, that is reabsorption. The filtrate has glucose, amino acids, and substance, water into it. So all those substances which are needed by the body, they are reabsorbed. So due to their reabsorption, only the waste products are left behind. Even you can see if there is deficiency in the blood, that is if there is low water potential into the blood, the water is also reabsorbed. So in this way, only left behind is the urine. Now the table 1.1 lists three of the components of blood plasma that enter the glomerulus in a healthy person. Complete table 1.1 by writing increased or decrease. So here you can see if the component is by writing increased or decreased, so we are moving towards the table. You may use each response once, more than once, or component of the blood plasma entering glomerulus. So the glucose, first of all, what is glucose is present in the blood plasma. Glucose is present in the filtrate at the same concentration as the blood plasma. It is very small molecule that can easily pass through the glomerulus. So glucose will be present in the uh, in in the component in the newly formed glomerulus. So it will present in the same. We will write a word same over here. Component in urine, it will not present in the urine in a healthy person. Not present in healthy person. It can be present in the person who is diabetic. So we are going to write about the healthy person. So it is present in healthy person. So secondly, the second you can see uh, in the urine, which is the large plasma proteins. So large plasma proteins are present in the blood plasma, but large plasma proteins are not present in the filtrate because they are too big to pass through the glomerulus filtrate barrier. Glucose small can pass easily, so it is present in the same, but they are large enough to pass through. So they are not present. If they are not present in the filtrate, so they will not present. Um, and the third is the urea. So urea will be present. So you can see urea is present in the urine. So, but its concentration is increased compared to blood plasma. Its concentration is low in the blood, but when in urine, its concentration is increased. Why it is increased? So first of all, it is present, same, same, increased. Because when water is reabsorbed, water will be reabsorbed, reabsorption. After ultrafiltration, the second step is the selective reabsorption. So what happens over here? Its concentration, you can say, is same in the urea into the glomerular filtrate. But when water is absorbed, water absorption takes place. Due to water absorption, its concentration is increased. So at that time, we can say that uh, it will be concentrated over here. Now we are moving towards the next question. Sweating during exercise can lead to a person's posterior pituitary gland and in the kidney. State the response to sweating that occurs in the posterior pituitary gland and the response that occurs in the kidney. Sweating. So sweating. What happens during sweating? During sweating, you can see sweating is uh, there you can say during exercise, exercise generates heat and sweating is the body's mechanism to cool down through evaporation. So Sweating produces heat which body cools down. It keeps the body to cool down. So loss of water and salt takes place, cool down, and loss of water and salts takes place during this sweating. So excessive sweating can lead to dehydration and electrolyte imbalance. Excessive sweating can lead to dehydration and electrolytic imbalance. So here answer question is state the response to sweating that occurs in the posterior pituitary gland and response that occurs in the kidney. So response to sweating involves the posterior pituitary gland. Posterior pituitary gland, you can say the gland which is present in the brain. It secretes a hormone, ADH, antidiuretic hormone. So the function of this, you can say antidiuretic hormone is body is dehydrating. Due to hydration, the body can have the serious issues. So what happens at that time when there is low water potential inside the body? So this antidiuretic hormone is secreted by the pituitary gland. So this antidiuretic hormones will affect on the process of excretion. At that time, extra water will be reabsorbed. Water reabsorption will take place, due to which water reabsorption there will be less urine. So due to less urine, the process of dehydration is reduced. Most of the water will be reabsorbed. So the function of this, you can say, antidiuretic hormones is to help to reabsorb. So in this way, we can say the kidneys also play a role in reabsorbing electrolytes. And what are those electrolytes? They are the sodium and potassium to maintain the proper balance in the body. So this is influenced by hormones like you can say uh, in we can also can say or we can briefly, I would like to say that the response to sweating involves the posterior pituitary gland releasing antidiuretic hormones to conserve water by reducing urine output and increasing water reabsorption in the kidneys. So we can say ADH is crucial for water balance to keep water balance in the body. So it is the primary hormones for responsible for regulating water. So this process is part of negative feedback loop. You know what about the negative feedback? So as blood volume returns to normal and blood solute concentration decreases, ADH secretion is also reduced. So it is in higher concentration at that time, it is reduced is that when the body is hydrating and the water potential is less at that time. So what happens? So water reabsorption takes place, and to keep the body healthy and in a balanced hydrostatic condition. So in this way, the antidiuretic hormones play a very crucial role. Now we are moving towards the second question, and second question is several different processes can affect allele frequencies in populations. Outline the process that may affect allele frequencies in wildlife populations. First of all, what are alleles? The genes are available in different forms. So the different forms of genes are called as alleles. So here question is outline the process that may affect allele frequencies in wildlife populations. So here I have written the answers. So I'm moving towards my answer which I have written. This is the key processes which can alter allele frequency. Which may affect allele frequencies in wildlife population. So the key processes which can alter allele frequencies in wildlife populations are natural selection. What is natural selection? Natural selection means the nature select the organisms which are best fit in the environment. Which are best fit environment, they are best adapted to the environmental changes. So when there is change in the environmental temperature, environmental, uh, you can say environment, in any kind of change which occur in the environment, the best adapted species, they will survive. They will best if they are. It means they are best fit in the environment. So this natural selection will help them, or nature will select those species. So in this way, their gene frequencies or their allele frequencies will be in, and the species which are not selected by the nature means they are not best adapted into the environment, so they will be eliminated. So key processes which can alter frequencies in wild are natural selection is the driving force for evolution. What is evolution? Evolution is the change with the passage of time. This is not sudden change, just like the mutations with the passage of time. So this is a change with the passage of time. So it can lead to an increase in the frequency of beneficial alleles. Beneficial alleles, these alleles which are selected by the nature, or the species which are selected by the nature. So they have beneficial alleles and a decrease in the frequency of harmful alleles. So the harmful alleles, they will not be, you can say, uh, selected by the nature, so they will be vanished from the earth, from the, you can say. So this, we can say, the natural selection is the driving force. So individuals with trait matter adapted to their environment are more likely to survive and reproduce, passing on those advantageous alleles to their offspring. So here we can give an example of peppered moth. Example, peppered moths, which were in the industrial England. So darker moths had a survival advantage due to the camouflage on shoot-covered trees, leading to an increase in the darker allele frequency, whereas the light-colored, dark-colored and light-colored. So in the industrial area, so they dark colors will be survived and light colors they will not survive because they would be very prominent on the dark environment or the shoot which has covered the trees or the other things. So dark peppered moths, they camouflage themselves so they will survive because there past environmental changes. So they will be selected by the nature, they will survive, whereas these light colored peppered moths, they will be vanished or they will be eliminated. And genetic drift can lead the loss of alleles on or the fixation of the others even they are not beneficial. So what is genetic drift? Random fluctuations in allele frequencies due to chance events, especially significant in small populations. So can this can lead to the loss of alleles or the fixation of others, even if they are not beneficial. So this is, you can see, uh, this genetic drift can randomly occur. Genetically, they will be different from one another. So they will have that this genetic drift, actually the random fluctuations in the frequencies of existing gene variants within a population. So it can lead to the loss of genetic variation and the fixation of certain alleles, especially in the smaller populations. So we can see it can cause changes in the allele frequencies over time, even in the absence of natural selection or other evolutionary forces. So it occurs when it occurs, the alleles passed on to the next generations are not always a perfect representation of the parental population's allele frequencies due to chance events like random sampling of gametes or random survival of individuals. And sometimes, some alleles might be lost entirely from the population, reducing the overall genetic diversity. So, in this way, we can say that these genetic drift can also uh, affect the allele frequencies in wildlife populations. And the other, the mutations are sudden change in the structure of the DNA or and the number of DNA can cause these mutations. So these are the ultimate source of all new genetic variations. Variations can occur, although mutations occur at a low rate. Mutations can be beneficial or lethal. Mutations are not always lethal. Sometimes they are beneficial. So we can say while mutations occur at a low rate, they are the ultimate source of all new genetic variations. Most mutations are neutral or harmful, but some can be beneficial and increase in frequency through natural selection. So if I would like to give an example of mutation, so we can say that a mutation in a plant makes it more resistant to a disease. So we can say, or you can say less resistance to a disease. So in this way, we can say the mutations can be harmful or they can be beneficial. In both ways, they affect the allele frequencies in wildlife populations. Non-random mating can alter genotype frequencies but do not directly change allele frequencies themselves. However, it can indirectly affect allele frequencies which individuals reproduce. So it can alter genotype frequencies, but we can say the types of this non-random mating can be assortative mating. Individuals with similar phenotypes mate more frequently, and you can say the individuals with dissimilar phenotypes mate more frequently. So it depends. As for example, I would like to give an example over here for as peacock with elaborate tails are more likely to attract mates, leading to an increase in the lead responsible for these traits. So we can say the populations understanding these processes is crucial for wildlife conservation. And there is one more thing which I would like to discuss, that is the climate change can alter selection pressure, favoring individuals with adaptation to new environmental conditions. So uh, climate also play a very role in changing the allele frequencies, and other hunting can significantly reduce population size, leading to bottleneck effect and loss of genetic diversity. So what is bottleneck effect? Word bottle, a sharp reduction in population size due to a catastrophic event or a natural disaster, you can say, resulting in a random subset of alleles survive. You can say just like a bottle, the frequency, it is reduced. it is reduced. So in this way, this is called as bottleneck effect. It reduces the allele frequency. And it can be due to a natural disaster. So in this way, I would like to say these are the different factors which can play a role in changing the allele frequency in different populations. Conservation, we can say by considering these factors, conservation biologists can develop strategies to maintain genetic diversity, ensure the long-term health of wildlife populations. Now, the next question is, the process of selective breeding changes allele frequencies in a population. Plant breeders use selective breeding to improve crop plants such as maize. Explain how selective breeding can be used to obtain a variety of maize where the plants are vigorous and of uniform. Selective breeding. Selective breeding, first of all, is a very, you can say, uh, diverse uh, process in which the different steps take place. First of all, they select a selection of parent plant, selection of parent plant. So in the first step, you can see farmers or breeders choose maize plants that exhibit the desired traits such as strong growth and uniform height. They need, they need the plants, the required plants. You can say the required plants are uh, should have these traits, and which traits they should be strong and uniform height. Uniform, uniform height. So this is the very first step that they will choose the parent plants. Secondly, crossbreeding. So selective plants, they will crossbred with one another. For what? Crossbred for what? For the beneficial traits in the offspring. So they will be crossbred. And thirdly, artificial selection. Artificial selection, they will select the offsprings that show the desired characteristics, then they will further breed them together. And then repeated breeding. Then repeated breeding. So repeatingly, they will breed them, and then multiple generations will be obtained and gradually increase in the frequency of a lead responsible for vigor, and then hybridization. So hybridization, hybrid maize varieties can be created by crossing two genetically distinct inbred lines to produce offsprings with hybrid vigor, which enhances growth and uniformity. So field testing can also, the new maize, next field testing. You have grown, you have grown, you have those after crossbreeding. So you have created, and then you need to test them. You need to test them. And when you will, you are going to test them, and then in this way you will be, uh, it will be very easy to find out actually which one is uh, continuously. So, so in this way, continuously they will select the plants which have favorable traits, and or you can say favorable and variable traits. So maize variety with uniform height and high vigor can be developed. So these are the different steps which can be a part of the process for selective breeding. And with the help of this selective breeding, they can select the plants with required traits, so with required qualities. So it is all about these two questions, number 1 and 2. um, Please like, share, and subscribe my channel. Thanks for watching. My email ID is given in the description. You can contact